So please use the question answer panel for asking questions. And if you need to speak, please raise your hand so that we will unmute you. And when you get a chance, please provide feedback on the details page. There is a link where you can provide feedback more than one time. So today we are going to present some of the work that we have been doing as part of the NIH Common Fund Data Ecosystem, which is essentially a consortium of 12 or more centers, large-scale centers, which are generating data of various types, such as metabolomics data, Biometabolomics Workbench, or I should rather say they are serving as repositories for various types of data. GTEx has data on gene expression in tissue. MotorPack has data of different types of related to exercise and nutrition in human and mouse, and so on. So the first presentation will be what we call as MetENP. It's a workflow for analyzing and interpreting targeted metabolomics data, followed by MetGENE, which helps extract metabolism and metabolomics workbench relevant information for one or more genes. Then I will do a quick walk through our GitHub repositories, followed by a presentation on metadata-based search on the Common Fund Data Ecosystem portal. So now going to the MetENP presentation. This work was largely done by Kumari Sonal Chaudhary with the help from several of us. I'm doing the presentation today. So MetENP is essentially a workflow which helps analyze metabolomics data and further interpret the results in the light of mechanistic information via pathways and networks. So the first step is to get the data, for example, for different metabolomics studies, identify what are the significant metabolites between two different conditions using the student's t-test and ANOVA. And then once you have the list of significant metabolites, one can perform metabolite class enrichment, metabolic pathway enrichment, and visualize those results, followed by further interpretation. For example, what are the reactions and enzymes which are associated with those metabolites, and what are the pathways in which those metabolites participate, and find out further information related to the enzymes in those reactions. MetENP is available as a R package, a Jupyter notebook, and also as a web resource. So today I'm going to give a demo of some of these resources. So how to use MetENP on the Metabolomics Workbench website? It's really simple. You go to the home page and then click on Analyze Studies. Then select a particular study. For example, in this case, I selected ST001140, which is a study of changes in the canine plasma lipidome after short and long-term exposure. Then click on the Submit button. On the resulting page, there are a number of tools listed, and Dr. Ointfai here already covered many of these tools yesterday. So today we are going to look at one of the tools, MetENP here. So you click on the MetENP web analysis link, where it allows you to select the analysis that you want to choose. For example, in this case, there are several types of analysis. Some measured phospholipids, others measured sphingolipids, etc. Then it provides a list of what are the different factors and conditions, so that you can decide with respect to which factor or combinations you want to perform the statistical significance testing. So for example, in this case, user selected treatment group, and then you click the Submit button below this. Then on the next page, it lets you select in each group, group 1 and group 2, one or more of the treatment conditions. For example, in this case, one could choose the in group 1 prednisolone, in the other one could choose the tetracosaptide. And then we have a number of parameters related to the statistical significance testing. For example, what method do you want to use for computing adjusted p-value, FDR or Benjamin-Hochberg, and then how to handle the missing data. There are 3 approaches. One is use half of the minimum value from the measurements, or remove the the samples with the NA value completely, or remove those samples where more than 50% values are NA. And then we have cutoff on the p-value, then a cutoff on the log2 fold change ratio. And then one could decide for further interpretation at what level you want to look at the classes, whether it's at the metabolite main class or subclass or superclass. And also you can decide how many minimum metabolites should be present in a pathway or class for it to be listed in the list of significant pathways of interest. One important thing I want to emphasize is that for part of the analysis, it is important to specify what the species is. For example, in this case, it's a dog. So it's the 3-letter code is CFA. And then you press the submit button. It takes usually a minute or two to do all the computations because there are several steps in this analysis. And then on the resulting page, you can click through these plots here. And these are just representative images, but when you click them, you get the actual PDF view for these results. For two of the plots, depending upon how many metabolites are listed or how many pathways are listed, the font size can be a little bit small or large. So we have the option to adjust those font sizes so that you can get a good quality view of these results. These results can be downloaded as individual files, or they are also available as a zip file, which also stores the values or the parameters that were selected during the analysis. So that is important for provenance purposes. I'm going to show just some schematic plots from the results. For example, here we can see the volcano plot where based on the cutoffs that we chose on the log2 fold change and p-value, we can see there are some metabolites such as phosphocholine, which are significantly upregulated in one of the conditions as compared to the other conditions that were selected, whereas there are several which are significantly downregulated. We can also look at with respect to broad categories such as based on subclasses, how many metabolites we have upregulated or downregulated in those. And here we, for the same class, we provide the count of upregulated and downregulated metabolites separately. We can look at the metabolite enrichment plot and also the metabolite pathways. So for example, in this network on the right side, it shows that it shows the pathways to which a particular metabolite might be participating in. We have some alternate views which show what are the metabolites that belong to different pathways or what are the pathways which are significant in our results. And similarly, this provides essentially an intersection view of the pathways and the metabolite classes. As Dr. Weinfeld showed yesterday, there are a number of other tools available, and he went in details over some of these tools, for example, volcano plot and related analysis. So I'm not going to cover that. MetENP tool can also be used to analyze users' own data, where they have the ability to upload their own data. So the way to do is to go to the main page and click on Analyze Studies. And at the bottom of that page, there is a link, Load and Analyze Your Own Data Set by MetENP. When you click on that, it takes you to a page where you can upload the file. And you need to select whether in the first column you have the sample names or you have the metabolite names. And once you select that, you click the Upload button here, and you will have the file uploaded. After that, pretty much all the steps are similar to what we saw earlier in the web version of MetENP. So I will not repeat those steps here. MetENP is also available as a Jupyter notebook, which is essentially backed by the corresponding R package. I will go over the GitHub repository for MetENP a little bit later in some detail. the GitHub page provides a detailed instructions about how to clone it on your local machine. We have tested it exhaustively on Linux operating system. So that's where I think one should try. One can try it on Windows as well, but we have not thoroughly tested it on the Windows platform. MetENP, for those who do not want to try by installing it on their local machine and then run it as a Jupyter notebook, it is also available on the My Binder page, where actually if you go to the GitHub page for MetENP, there's a link that directly takes to My Binder, where you can essentially launch the notebook in a container and see how it works. Sometimes launching the notebook the first time can take a bit of time. So because it might be trying to build the container based on initial image, so that can take some time. So if it shows any errors, just give it some time, and sometime just restarting takes care of any issue. Another tool that we have is called as MetNet, which is essentially a subset of the MetENP workflow that I just described. And the only distinction is that with MetENP, usually the starting point is a metabolomics study ID or a data matrix with the targeted metabolomics data, where we first identify the significant metabolites, and then mechanistically interpret those results in terms of pathways, networks, and related reactions and enzymes. The purpose of MetNet is if the user has already a list of metabolites of interest based on any other study, or they carried out, they identified the list of significant metabolites based on some other analysis, then starting from that point, one can perform the interpretation of those list of metabolites using this MetENP MetNet pipeline. So MetNet is available as a REST API or smart API. However, it is actually best used as an integral part of another workflow or tool, such as Playbook Workflow Builder tool, which I will go in more details tomorrow. So with this, I conclude the presentation part. Now I will go briefly to the GitHub page for MetENP and also to the Metabolomics Workbench website and provide a demo of MetENP. So let me go to the Metabolomics Workbench website. We go to the Analyze Studies page, and from here we can choose the study. For example, let's do study 915. It's a study about progression of NAFLD and biomarkers associated with that. So I will go to the TNP section here. It takes a little bit of time to pull out all the information associated with the study. Here one can choose which analysis one wants to try. In this case, I'm going to select all. And here I can choose which column from the factors information table I want to use for the t-test. So in this case, I'm going to select the diagnosis column. Now here I have the ability to select one or more conditions from this list to be included in the group 1 or group 2. So in this case, I'm going to choose the group 1 as cirrhosis and group 2 as normal, and because usually we see maximum difference between those two conditions. For the adjusted p-value, we can choose the examining hyper. For handling missing data, let's say we want to remove NA values. Usually, I try a higher p-value cutoff so that I get a relatively larger number of metabolites, and then later I can use a lower p-value cutoff if I'm getting sufficient number of metabolites. In this case, I'm going to essentially not apply any cutoff on the log to fold change value. And I want to see all pathways which have one or more significant metabolites. In this case, it's human data, which is the code for the species. When we click on the submit button, one can do some additional analysis related to K pathways. However, that can take a little bit longer, so I'm not going to show that in this demo here. You click on the submit button, and it takes about 2 minutes to do all the calculations. So while those calculations are happening, I can go to the GitHub page for MetENP, where we have provided extensive details about how to install it locally on your machine and how to run it. So essentially, it starts with the cloning the repository on your local machine following this git clone command. And in order to install MetENP as a package on your local machine, you need to have DevTools installed. If you have not already installed, follow this command. So it is expected that the user who wants to use MetENP as a Jupyter notebook, they have good familiarity with the basic R commands to follow through these instructions. I will skip some of these ones. Assuming that you have Jupyter installed, if not, one can follow the details in this section here. Skipping that, I will go to the section how to install MetENP R package through DevTools in user's account, so that you don't need to worry about system administrator privileges. I have described it in extensive details, and if you have ever encounter any issues, please contact us. And in the later part, we do have instructions for some advanced features. So in case you want to make any changes in one of those functions, or if you want to add additional functionality yourself. Now here, it also provides a link to launch MetENP on my binder. So going back to the results from the analysis that we did on the MetaBolomics website, here we can click on the plots to open a new tab. It's essentially a PDF file, which can be saved in different formats or printed and downloaded. And then we have the count of metabolites. We have results in terms of the enrichment of different subclasses of metabolites. This provides a view of the metabolic networks, showing what are the metabolic pathways in which these metabolites belong to. And we have the ability to download these results. So with that, I think I have covered most of the steps that I wanted to show on MetENP. Going to this, I can show how it looks on my binder. So I launched it some time back, so that if it needed to recreate the container, it will do that, because sometimes that can take 15-20 minutes or even slightly more. So this is essentially a Jupyter notebook where you can follow through all these steps. If you need to, the good thing about Jupyter notebook is that if you want to make some changes in the analysis, for example, if you want to change one of the parameters related to the t-test or something else, or if you want to select a subset of the analysis to be included in your statistical analysis, then one can do that through Jupyter notebook, following all these instructions and then changing the commands locally. Essentially, this provides all the same results that we saw earlier, but with the ability to make changes yourself and test out various things. Sometimes I add quite a bit of debug lines so that if there are any issues, I will get all the information. So Jupyter notebook is helpful from that point as well. With that, I conclude this part of the presentation, and we'll be happy to take any questions. If not, I will hand over to Dr. Sumana Srinivasan to talk about MetGENE. And if there are any questions related to this, you can ask later too. And a little bit later, we have a hands-on session where you can try some of these. And if you encounter any issues, please ask questions.
Good morning, everybody. My name is Sumana Srinivasan, and I'm going to be presenting today a tool called MetGENE. Thus far, you have heard about how to access data and studies in Metabolomics Workbench and analyze these studies through the extensive talk Dr. Oenfe he gave yesterday.
Sumana, I'm sorry, let me interrupt you here. Do you want to go to presentation mode?
Oh, sorry. Thank you. I was about to do that. Yeah. And just before me, Mano talked about how to do analysis starting from a set of metabolites in MetNet or through study IDs using MetENP. This particular tool provides you a gene-centric metabolomics information retrieval. And why would that be interesting? So current tools focus only on gene metabolite linkage. But here, MetGENE focuses on linking genes to metabolomic studies. Let's look at a few applications where this would be quite interesting. Consider the case of personalized nutrition, where a clinician needs to identify studies for clients with metabolic conditions to tailor their nutritional plans. So one could look at a particular gene, such as lipoprotein lipase, to see which metabolites are overexpressed when this gene is overexpressed, and what effects they have on their diet, and such studies will help them tailor their nutritional plans. Further, in biomarkers research, one would want to identify studies linking metabolic genes to specific disease biomarkers such as diabetes or cancer. For example, the variants in GCK or the glucose kinases has significantly higher in fasting blood glucose levels, and it reduces the insulin sensitivity and increases insulin resistance compared to controls. And this could be a biomarker for diabetes. One could also measure environmental stress response to see what impact these environmental stressors have on the metabolome and cause the gene changes in gene expression. Example is a gene called cytochrome P450-1A1, CYP1A1, which is involved in the metabolism of pollutants. And last but not the least, the pharmacogenomic profiling to connect metabolic genes with metabolomic responses for various drug treatments and clinical studies. An example would be a CYP2D6 in the treatment of breast cancer. This metabolizes tamoxifen to an active form called endoxifen, and this can be used for drug-related studies. So having motivated the need for linking genes to metabolomic studies, now let us see how the tool works. So as I mentioned, it is a knowledge-based tool focused on genes encoding proteins associated with only metabolites. We are not covering the entire gene set, but only focusing on the metabolic genes. I say a knowledge-based tool because the way the metabolomic studies is accessed when a gene term is given is that gene participates in pathways. As I can see, let me just turn on my pointer. Give me a second. Yeah. So a gene participates in pathways. Pathways contain reactions. Reactions contain metabolites, and metabolites are part of metabolomic studies. So that is the way MetGENE aggregates information and provides the user, given a gene, all the pathways, reactions, metabolites, and metabolomic studies. And as I said, we are only focusing on those genes that encode proteins that are directly associated with metabolites. And here on the rightmost side, we have the number of currently the MetGENE supports 115,770 metabolites, 3,932 reactions, 6,196 pathways, and 406,048 studies. A paper based on this tool was published recently in GigaScience, and we will share a link to this paper for people who are interested to know more about the tool later. So what are the key features of MetGENE? It is a centralized information access where it retrieves data from different standard repositories for genes, for pathways, for reactions, metabolites, and studies. It displays all the information on a single dashboard and has tabs for intuitive access and the tables available for easy download both in JSON and CSV formats. And wherever possible, information is hyperlinked to relevant websites, so all the information is available at the fingertips for the user. It also provides contextual filtering, where filter is based on species, anatomy, and condition. The species you can choose. Currently supported are humans, mouse, and rat, and a whole bunch of anatomy based on the samples that are there in the metabolomic workbench, and conditions, both disease or phenotype, like diabetes or cancer, or phenotypes such as obesity, as an example. And this is an open source tool. The source code is available. And also the APIs are available both with REST and Smart API formats. I'll demonstrate that towards the end of my talk. And the tool can be accessed in this hyperlink. This will also be posted in the chat for your access. So the MetGENE architecture comprises of a front end comprising of PHP and JavaScript. At the back end is a whole bunch of our code as a package, and this can, through via REST API, talks to backend databases such as the MetaBolomicsWorkbench database to obtain the metabolomic study information and study IDs, RefMet and MetStat information, KEGG database for reaction information, KEGG reaction IDs and names, pathway database links to common pathway databases such as Pathway Commons, Reactome, Wiki Pathways, KEGG, etc. And gene information obtained from gene databases with links to NCBI, gene cards, Ensemble, UniProt, Marvel, etc. So let me go through a quick demo using my slides, and then I will go to the actual website. So this is a screenshot of the MetGENE query interface, and it provides you a text box where you can give a list of genes or a single gene. You can choose whether the gene ID type is a symbol, or you can even type out a UniProt ID or an Ensemble ID and then pick up the appropriate gene ID type. Then you can use the filters, as I mentioned, the organism filters, anatomy filters, and disease phenotype filters. Here I've used HK1 and ALDOB as an example. And then we have since these are symbols, we have chosen symbol. Then let's look at the human as an organism filter. Let's use blood anatomy. And here we are using metabolic disorder. The disease menu is twofold, because we did not want to present you a laundry list of diseases, and they're organized based on what type of disorders they are, and beneath that would be the disease that you may want to search for. And as soon as you hit submit, the left-hand side is the landing page. It gives you a nice graphic about the context, like what are the filters that you have chosen, what are the genes, and then on the right-hand side, what are the tabs that you can access. You can access through these buttons as well as through the tabs that you see above. I just want to quickly bring you to the summary. The summary tab, when you click on it, gives you the summary of how many pathways, reactions, and metabolites and studies are pertaining to this, but the set of genes that you have chosen. It gives you both a nice graphic in terms of a pie chart, as well as a table which can be downloaded both in the JSON and in the CSV formats. So when you click on Genes tab, let's say the Genes tab is highlighted in pink to give you the context, and it gives you the gene information for all the genes that you have chosen. So you have this as symbol, and all the gene databases that are supported for gene information are KEGG, gene cards, NCBI, Ensemble, UniProt, and Marvel. And clicking on one of those links will take you to the appropriate page in the gene cards as a separate tab, so that the user can look at these genes in more detail. And you can use this to JSON or to CSV to download the table in the JSON or CSV formats. Further, when you click on the pathway tab, again, the pathway tab is highlighted for you. And again, here you have all the pathway information for Pathway Commons, Reactome, KEGG, and Wiki Pathways. And clicking on, say, an ALDOB under Pathway Commons will take you to the Pathway Commons page, where you can look at more information on the pathways related to ALDOB. Similarly, the Reactome pathways. Next, clicking on the reactions will bring you to the reactions tab. And here you find all the reactions associated with both HK1 as well as ALDOB as two separate tables. And each of these tables contain the KEGG reaction ID, the KEGG reaction name, and the KEGG reaction equation. for your perusal. And then clicking on the KEGG reaction ID would directly take you to the KEGG reaction page for that particular reaction, where more details can be seen. And again, as always, these tables can be downloaded for further download processing, both in JSON and CSV formats, as you can see in the right-hand bottom of the picture. Further, clicking on the metabolites page, you get to see the metabolites associated for all those reactions, based for all those genes. So HK1 and ALDOB has two separate tables and all their metabolites. Here you have the KEGG compound ID on the left-hand side, the RefMet name, which Owen described in detail yesterday, is provided as hyperlinks. Clicking on it will take you to the RefMet page. KEGG reaction IDs are provided again, and clicking on it will take you to the KEGG page, and the MetStat link associated with that metabolite. So if you say, so again, metabolites tab is highlighted for you. And if you click on glucose, that will show you to the RefMet page here. The RefMet compound details are all given. And clicking on the KEGG ID will take you to the KEGG compound page as it is given here. And clicking on the MetStat link will take you to the MetStat page here. And you can see that for glucose, the median value is more than 100, because we have chosen the filter of the disease as diabetes, and obviously the glucose levels are higher. So this takes you to the MetStat page related to glucose. And again, these tables can be downloaded in JSON or CSV formats. Then we have the studies page, and here you get the consolidated studies for all the list of metabolites that we have for both of these genes. And not only, so clicking on a study ID, you can directly get access to the summary of the study in the MetaBolomicsWorkbench page. And so hovering on a study ID will also give you the title of the study, so that you can look at those studies and only click on those of which that you're interested in. And apart from that, we have an ability to pick different metabolites, groups of metabolites, and combine studies if you would like to download those studies separately for your downstream processing, as shown here. So this is a combined studies page. With that, I completed the demo on my slides. I will just now move on to the actual demo. Let me just show you the GitHub page for MetGENE, which Mano will elaborate much later. And it also has some REST-based API for you. And control click. So you can choose for reactions. If you specify the species and the gene ID symbol, it can give you a REST-based interface for accessing all this information. We also have registered this in the Smart API, and here you have you can use the same REST-like interface, but you can actually specify the strings here in a more user-friendly way. You can give that name of the species ID, the gene ID type, and gene ID, and the type of what type of output that you expect. And when you execute that, you get a JSON response here, and this link will also be shared. Okay, let me just
Okay. My question was so in this presentation, you showed how you can start with the gene and then identify the metabolites that come out of it and all that. So can you do the reverse, in the sense that if we have a set of metabolites that people have identified, to go back and see which genes are?
Yeah, that is already provided. Owen covered it in yesterday's talk, that you could go access genes and pathways through the Metabolomics Workbench interface.
I see.
So what was missing was the gene to metabolomics link, which is actually the MetGENE bridges that gap.
I see. Okay.
And also the MetENP pipeline, at least part of that pipeline, especially the MetNet part, that exactly addresses that. If you have a list of metabolites that are important to you for one reason or another, then you can use that pipeline to find out what are the related genes and pathways, etc.
Okay. Okay. Thank you.
Interrupting. Okay, I started with a gene called RPE, which is another metabolic gene. And here is the landing page for RPE. And let's go to the genes tab. And while this loads, on the right-hand side, it gives you all the details about the RPE gene. And here we have the gene tab, which, as I mentioned, the genes tab is highlighted. And here you can control click on it, open the link in new tab. It takes you to the appropriate KEGG page pertaining to the gene RPE. And next, we can go to the, similarly for gene cards and NCBI. Let's go to the pathways. And again, let's probably choose the reactome, open it in a new tab. And it takes you to the reactome page corresponding to that pathways that RPE is involved in, followed by the reactions, which is actually goes to the KEGG database to obtain all the reaction ID. There's only one reaction pertaining to this particular gene. And here is the link to the KEGG page corresponding to that particular reaction. So you can look at the reaction in this particular KEGG page. Further, we can go down to see what are the metabolites that are there in these reactions, which will be presented to you in the form of a table. And so this takes you to the KEGG compound ID, as I was mentioning. So you can go to the KEGG compound ID for that particular metabolite, D-ribulose 5-phosphate. And let's go back here. And if you want to know what is the RefMet name, this is going to take you to the corresponding RefMet information page. So this is the RefMet details page. I think I'm going to close some of the tabs, maybe tab overload. I'm just opening it in new tab because I do not want to lose the context, but it can open. Then further, this is again the KEGG reaction ID. And we can go to the MetStat link corresponding to the this particular metabolite. And here is the details of the MetStat details for that particular metabolite. And let's say now if I say to JSON, it's going to download a JSON file, and this contains a JSON output. I'm not going to open it just in the interest of time. And further going down to the studies, it gives all the studies associated in the Metabolomics Workbench. For this particular thing, there's no studies found for the other metabolite. So hovering on the, as I mentioned, hovering on the link will give you the title of the study, as I mentioned before. And clicking on this will take you to that particular study in the Metabolomics Workbench. And since there's only one study, the combining studies doesn't make sense here. And again, we have the summary page here to show you the summary of how many pathways, reactions, metabolites, and studies are corresponding to this particular gene. So that is the way, and you can go, when you reset the query button, you can reset and type any other gene that you would like to search and go back to searching the MetGENE interface, and it'll take you through the same thing. With that, I conclude my session, and I just want to give a quick introduction to the next session where my colleague Shiva Ramakrishnan is going to present going beyond the Metabolomics Workbench. So,
all right, thank you, Sumana. So now we have about 40 minutes session to try some of these tools out, and then you may have some questions or suggestions. So do you see the GitHub page for Metabolomics Workbench?
Yes, Manu.
Okay. So we have several of these tools also listed at our GitHub page, that is github.com Metabolomics Workbench. So I will briefly go over some of those, starting with the RefMet. I think I already have that one. Okay. Okay. So as Dr. Weinstein, he described yesterday, RefMet is essentially a tool which can help you identify or find the RefMet name for any metabolite name or list of names that you may be looking to convert into RefMet name. So essentially, it's a R-based package which you can either install after cloning, or you can just source those R files and then use it in your program. So you can install it using the devtools command. You can specify where you want it to be installed so that you don't have to worry about administrative privileges on your machine. Then you do need to, sometimes you may have to include the path where you installed this tool. And then here we have some examples of how to use this tool. For example, you can read the file where you have the list of your metabolites and then follow through the series of commands to convert the name that you have into corresponding RefMet name. In some cases, it may remain the same, or in some cases, it might be somewhat different as Dr. Weinstein described yesterday in more detail. It also provides some visualizations about of the list that you submitted to which different, what are the different classes to which they correspond to. For example, here it is describing in terms of the distribution with respect to the main classes. You can also choose subclass or superclass here. You need to compute the expected enrollment values for an adapt of interest. You can follow these additional commands like adapt tab to do that computation. So I will move on to the next repository, that is MetENP. So as I described earlier, MetENP is available as a R package and a Jupyter notebook, and also it can be accessed on MyBinder. And this repository has all the necessary files to serve in either modes. What I have done here is given extensive detail about how it can be installed on your local machine, depending upon what you have already installed. For example, you can clone the repository and then install it using the DevTools package. You may have to install a number of necessary packages before it works successfully, depending upon what other packages you have already installed. For example, it makes GetRest, GetGraph, and PathView package. One thing to note is, as such, this repository follows the MIT license, so you are free to use it in any way you want, including making your own tools based on this. Something to keep in mind that it uses the CAG-related resources. So they do have some restrictions in terms of in what way you can use their resources. So it is important to understand their terms and conditions before you use it for a specific purpose. For example, if you want to use it for your own research purposes, that is free. But if you want to provide a service based on this, such as server, some web content based on this, then it's important that you go through their precise terms and conditions and make sure that you understand them and not violating your rules. It can also be installed through Anaconda. I will not go into the details of that, but it is really a simple step. If one actually does not want to install it as a package, then after cloning the repository, you can just load source various files and then just load the appropriate libraries, and that should also work. As I have shown here. if one needs to install Jupyter package, then one can follow the instruction, detailed instruction in this section. If you already have Jupyter package installed and it's working properly, then you can skip this section and go to installing the MetENP package. Here I have described how to install it inside a folder such as your home directory, such as the .local folder in your home directory, as that is a common location for installing the user software and packages. Alternatively, if you want to install it in some other locations, for example, in whatever folder you are, if you want to install it there, you can use this set of commands. And again, it is understood that some familiarity with our commands and packages is necessary in order to follow through these instructions. Once you have installed it successfully, you can check it by including the path to the install location and then issuing the library MetENP command. And after that, you can you should go to the folder called Vignette, where there are a number of Python notebooks, and you can get the Jupyter command while doing there. This IP address is the IP address of your machine, and assuming that the port 8080 is open on your system, you can use this port. Or if there is any other port that has been opened by your system administrator or yourself, then you can use that port and follow the instructions that you get on the screen after you get this. I provided some instructions about some advanced features, such as if you want to change the functionality in one of the functions, or if you want to add additional functionality, then you can follow through these steps to modify those files and then test it out. With this, I will go to the next repository, which is MetGENE, that Dr. Srinivasa covered in our presentation. So even here we have provided all the necessary files, and you can install it on your local machine by cloning it, and then run some basic steps such as extracting the pathway or reaction or metabolite information for a given for a single or more than one gene. We have listed all the commands, necessary commands, how to run that. I do want to emphasize on the conditions imposed by the CAG license that you can use it freely for your personal purposes, but if you want to serve web content based on this, then it's important to go through their terms and conditions and make sure that you do not violate anything. What I want to cover here is that MetGENE, much of the functionality of MetGENE is also available as a REST API, a REST interface. For example, as we can see in this URL, we have the ability to specify the gene symbol. We can specify the anatomy if we want to, or leave that as NA, but the field must represent. Similarly, for other fields such as disease phenotype, and then we can decide whether the output we want it as a text format or a JSON format. So, for example, if I follow this link, it takes a few seconds to do all the computations and then present the results in JSON format. If you want to see the raw data, our browser allows us to see this. The browser has rendered it in a more readable format here. Similarly, we can look at the functionality that we saw on the metabolites tab in the demo that Dr. Srinivasa provided. I can take an example of two genes where we are specifying that these are gene symbols. In this case, we are looking at the disease diabetes. This might take a little bit longer, as it is going through the steps of identifying all the other gene IDs and then finding the reactions and then providing the metabolite information. So the benefit of having the REST API is that if you are developing your own tools, and then if you want to include this functionality, you can follow this style to construct these URLs and then fetch the data from there in your program, whether it's R program or some other Python or some other language. Let me take an example of the summary view. So in this case, on our servers, we have a timeout limit of about 5 minutes. So if some gene list of genes takes more time than that, it's possible that it will time out. So that is something to keep in mind. Sometimes you may want to submit just one gene at a time if it is taking a lot of time. For example, in this case, we submitted 2 genes and it is providing information, summary information for the JSON output. Going back to the original list of repositories, we have a few other ones, such as the gene ID conversion tool. I don't see here listed, but that's actually used on the back end in the MetGENE package. So if you have any suggestions or if you notice any issue, you have the ability to go to the respective repository and click on this tab, and then you will be able to create an issue, which we will be notified and we will try to address those. Something we have found that repositories such as MetGENE and MetENP. So that essentially what I had in mind for a quick walkthrough of our GitHub repositories. If there are any questions, please raise your hand and we will try to address those. Panelists can monitor if anybody has raised their hands, and if so, you can unmute them so that they can ask any question. Okay, I will start sharing.
It's visible, Shiva.
Thanks for confirming, Sonu. So, good day, everyone. I'm Shivaramakrishna Srinivasan, and I will be presenting this talk titled A Metadata-Based Query Interface for Enhanced Usability and Integration of Common Fund Data. So the goal of this presentation is to really showcase a novel interface that enables biomedical researchers to discover and access common fund data across all DCCs from a single point of access. And the way this works is through metadata-based queries. Before I begin my talk and delve into the details, I'd like to acknowledge Avimayan's group at the ICANN School of Medicine at Mount Sinai and the Subramaniam Lab team at UCSD. It is a joint effort that has helped build this tool, and I'll begin my talk. So, what is the CFDE? Just to double down on what we've heard so far from Manu and Sumana and the other presenters from yesterday. The CFDE came about to enable the broad use of common fund data to accelerate discovery in 2 major ways. The first is to make common fund data findable, accessible, interoperable, and reusable according to FAIR guidelines. And the second is to enable researchers to discover this data across the data coordinating centers so that they could ask scientific and clinical questions from a single point of access. For example, the different DCCs are shown over here, And each of these DCCs have their unique experiments and data. For example, the Human Microbiome Project contains rRNA and whole genome shotgun sequencing data that provides information about the microbiome in humans. The 4D nucleome, on the other hand, contains data about chromatin topology, which explores the organization of DNA in the nucleus. GTEx, on the other hand, provides tissue and cell-specific gene expression data. And MotorPack provides other forms of data that are primarily useful for understanding the effects of physical activity and exercise in humans. So as you can see here, the kinds of data that we have from each of these DCCs is diverse and very heterogeneous, and it warrants a need for a unifying model. And that is exactly what is described by the cross-cut metadata or the C2M2 model. This essentially enables data sharing across these DCCs by being a minimal and common metadata model that describes experimental data from each of these common fund projects. What it also does is serves as a harmonized model across these standard ontologies. So the figure you see on the right over here, essentially this is a condensed version of the C2M2 schema, which consists of over 49 tables. And all year round, DCC submit metadata to the DRC, which is around 25 gigabytes. And I must credit the development of the cross-cut metadata model to Owen White and his team. And if you're interested, and the slides will be shared later after the workshop ends, you can take a look at this particular reference that really describes how the C2M2 came to be. So to also provide a short explanation of what exactly and how intricately the schema of the C2M2 schema is constructed, I want to talk about how DCCs typically tend to organize their studies into projects, and how projects are associated with subjects. And each subject consists of some specific taxonomy information and are directly and also consist of information related to genes and proteins and phenotypes, diseases, and substances. Now, biosamples, on the other hand, are also directly related to subjects as they are sampled from them. And the way biosamples are obtained are via assays that ultimately generate files which contain a few attributes such as file formats, data types, and are generated by certain kinds of analyses. And eventually, projects end up containing files directly, and there is a intricate relationship between each one of these tables. And to give further examples of how these each of these tables are related, I'll explain further along in the demo of the actual DRC portal. But it is worth noting that each of these tables are constructed based on real experiments and are quite useful in helping us come up with this common metadata model. And it's really a unique effort that has come into place. Moving on, I'll talk a little bit about how a user might want to discover metadata through the CFDE DRC portal. So let us consider the use case where a user has a question as to whether statins have adverse effects on a patient post-treatment, particularly with respect to diabetes. So the user would perform a query across these DCCs and may do so in two ways. The first way is through the metadata query engine, which shall be the focus of this talk. And this provides an intuitive navigation without prior knowledge of the underlying schema. So the slide that I explained to you just before this is not something that is necessarily presented to the user, and a much more simplified version of what is present in the schema is what the user sees and is easy to grasp. We also provide context-sensitive filtering to help users refine searches based on relevant criteria. And we do this through a user-friendly display and by seamlessly integrating data and also providing the ability to export data to the cloud. And there are multiple cloud service providers from which data can be downloaded. So we primarily allow the export of data through the CWIC. The second arm of querying is via the process data query engine, which enables the discovery and the integrated analysis of the process data using a collection of tools and the cloud workspaces that you see here. And this can also automatically generate human readable reports from the analyses. And these can further be analyzed downstream through tools such as the Playbook Workflow Builder, which my colleague Mano will speak about later in this workshop. And this is really how a user workflow would take place when it comes to the metadatabase and the process data query engine. But for the focus, for the interest of time, and the main focus of this talk shall be the metadata query engine. Moving on, I would like to talk a little bit about the architecture of our new platform. It consists of a front end, a middleware, and a back end. And we have developed our front end using React, which contains multiple React components for a modern and interactive user interface. And it also provides an enhanced user experience and easier navigation through the next framework. It's also a researcher-friendly representation of the C2M2 schema. And as an internal benefit, we are able to develop in TypeScript, and this essentially allows us to develop type check code. For the middleware, we use a tool called Prisma, which provides a bidirectional interface to the database. And using the Prisma client for data retrieval, updating our schema, and manipulating our data has helped really smoothen the workflow of interfacing with our backend. And the added benefit of using Prisma is we can write our queries to the database directly in React. And speaking about the backend database, we have two prongs associated with, we have a two-pronged approach associated with storing our data. We have a reliable Postgres database, which can help us query quickly in SQL. And we also store our data in Amazon Web Services. Then this acts as an extensible and robust storage mechanism and service. And we also have a Neo4j graph database that enables an even more intuitive and interactive query system via a graphical query language, which my colleagues Srini Ramachandran and Ethan Sanchez will talk about later in this workshop. Before I jump into a live demo of the CFDE Data Portal, I want to pause for a minute and consider some use cases that might showcase the power and the diversity as well as the novelty of the CFDE Data Portal. Consider biomedical researchers who wish to identify different kinds of data associated with type 2 diabetes. On the other hand, there might be bacteriologists who wish to understand the role of microbial resistance in the human microbiota. And they may be looking for a very specific kind of data type. Similarly, oncologists may wish to look at microRNA data that is associated with pancreatic cancer. All three of these kinds of users can access the kind of data that they want through the CFDE Data Portal. And we provide a variety of data types that these users can pick and choose from, such as DNA sequences, count matrices, protein sequences, gene expression profiles, images, RNA-seq data, mass spec data, as well as sequence variants and feature tables. These are representative of the many more kinds of data types that we have in the CFDE Data Portal that is accessible today. So I'd like to speak a little bit more about the features of our metadata-based search. So, and this is this shall be shown as a live demo shortly on data.cfde.cloud. So this is what the main search page looks like. And you have a very clean and intuitive search bar here that allows you to perform full text search across not just the C2M2 metadata query, but also metadata search, but also process data and other databases. So on the left, you see here, when the results page pops up, you have the ability to switch between tabs here in order to identify and really narrow down your queries. And you can also select individual records here shown in this neatly organized results page, which is also paginated based on your requirement and convenience. And you can choose individual records and download them into a JSON format as shown here. And you can also filter based on different categories, including disease, species, anatomy, gene, protein, compound, data types, and the common fund program that you prefer. And you can get data specific to these attributes on your main search results page. And you can also choose to toggle the number of records you see per page. And this also provides a more granular level of control when it comes to viewing the results on your main search page. Now that you apply filters, you can also provide a much more granular and fine-grained control when it comes to these cascaded filters. You can, as you can see here, the filters are organized neatly by the category that they belong to. And you can also do a one-shot reset of these filters and redo your query from scratch again. Or you can deselect each of these tags to also quickly get to a very specific filter set that you want. And once you're satisfied with your filters, you can always hit the view icon here to open up the particular record of interest to you. And this opens up a rich and informative records page that shows the summary of the project, which includes details like the project description, ID, URL, taxonomy, sample source, and other disease and data type related information. You can also see the assets that are associated with the project, and this includes information about the biosamples associated, the subjects associated with the project, as well as the files that are associated with the project. And really, the primary goal of each of these record pages is to give you access to the files at the end of the day. So there is also a view where you can access these files directly, and you can download them as JSONs, or also copy a DRS bundle, which gives you direct access to the files, downloadable links for the files, which I'll be showing you now. So without further ado, I'm going to skip right into the demo. We have two links for the demo today. So we have our main public server, which is data.cfde.cloud. And we also have a local server that runs on ngrok. And for those who are interested in using either one of these two, you can use this username and password, which Manu will be sharing later in order to see this. So I'm going to jump right in and open up one of these links. Is my browser visible?
Yeah, we can see it. And I shared the ID and password for the ngrok page in the chat window.
All right, sounds good. So you can I'm going to enter a query here, such as liver, for example, and I hit search, and this gives me a view, a tab-based view of the different kinds of results that are found here. And I'm going to be focusing mainly on the cross-cut metadata aspect of the portal, but uh, there are other tabs such as the process data tab, results from motor pack, gene sets, anatomy, file information, and so much more. So as far as the cross-cut metadata uh, interface goes, I'm going to try and filter this based on specific criteria to show you the exact power and capabilities of the data portal. As you can see here, we have the list of records shown here, and they're neatly organized in this tabular view, and based on the DCC that it's associated to, the project name, other attributes such as species, disease, the source of the sample, and the data type, and the assets associated with the project. I should also mention that you can go directly to the link associated with this project by clicking on the value here under the project name, and this shall take you directly to the study or the publication that is associated with that project. You can also toggle the display per page to whatever number you want, so you can get more, you can have more fine-grained control of the number of records you see per page. And there is also, so as you can see, I changed the display per page to 50 from 10, and I have an even more extensive view of the number of records that I have here. And just to demonstrate the ability of these cascading filters, I'm going to try and first sort this out by the kind of common fund program that I'm interested in. So for now, I think I'm going to try and select the UCSD-MetabolomicsWorkbench as the common fund program that I'm looking for data from. And this yields all the results, which are 266 in number, associated with UCSD's Metabolomics Workbench. Now, I think it's an interesting idea to try and stack certain filters. So I think I'd like to view records related to Homo sapiens as well as mus musculus or mice. And this easily yields these filters, and it's right above my table view so that it's easy to figure out what tags I can select and deselect. And as far as anatomy goes, I'm also going to try and type in something like blood serum here in order to get records that are related to, that are sampled only from the serum of the blood. And this gives me a results view that that yields records only related to blood serum. So what's also interesting here with the cascading filters is that they're all neatly alphabetized, and you can always start typing in a few key letters, and it auto-completes and gives you only the search, that gives you only the filter that you want to view. Simultaneously, I can also, as I explained before in my slides, I can also deselect some of these filters, and I can also choose to reset them all at once. But I'm not going to reset them now. I'm going to maybe just deselect mus musculus and identify only records that are related to Homo sapiens, for example. So I can choose to download a few of these records as JSON, and this will allow me to save it to my desktop. And this, and I've already kept this downloaded, and you can see how the main search results page and the records that have been selected from there can be easily viewed through any text editor of your choice. You can identify, you can download this data and use it for further analysis. So I'm interested in understanding what's present here in this second record. So I'm going to go ahead and open it up here. And as you can see, this is an interesting project about the identification of pre-diagnostic lipid sets associated with liver carcinoma. And this is a study by the Metabolomics Workbench. And as you can see here, there is a short blurb that describes this project, and it consists of other project-related information. Any link that you see over here is a hyperlink, and it will take you directly to the associated relevant page. For example, the project URL should take you directly to the Metabolomics Workbench study over here, and the link under Homo sapiens should take you to the NCBI taxonomy term for Homo sapiens, and you can learn more about that if you wish to. Similarly, the sample source also takes you straight to the EBI ontology search link, and you can learn more about the sample source that is associated with that project record. And you can do the same for the disease as well as the data type information. And these take you to standard links such as Bioportal and disease ontology to learn more about each of these fields associated with the record. And well, scrolling further down, you see all the information associated with the assets that are relevant to the project. And there are 438 biosamples here that are consist of a biosample that are defined by a biosample ID and a persistent ID or a access URL that you can look at. And you can also perform a download of this particular table and view it later in your own text editor. Similarly, there is also subject information and information of files associated with respect to this particular project. And here is where we also help you download the links that are directly associated with these files so that they can be put into a program like CURL or wget, and you can download these directly onto your system. So if I were to select all the files here, I can either download this as a JSON, or I can hit this button to copy these particular access URLs that are associated with the files. And I can go ahead and paste them in my favorite editor and essentially save this as a file, and I can download these through any batch process, like I mentioned, such as CURL or wget. And that's all I have for the demo. And I'm happy to take
Shiva,
questions.
if I may add, so these
Yeah.
DRS bundles can be directly copied into cloud workspaces, and it can be copied into any cloud workspace, AWS, Kavatica, or CWIC. It's not restricted only to CWIC. So these, but copy DRS bundle copies it into a clipboard, and it can be directly pasted onto the cloud workspace. That's all I wanted to add. Yeah.
So I totally invite all of you to try out this tool. As I said before, there are 2 different links here that you can try it out from. You can do it without a sign-on option from data.cfde.cloud, which is public right now, or you can try out this tool using the second ngrok link, which would require this username and password, which Mano has posted. So I'm happy to take any questions now, and please feel free to try out this tool. And while this is still in production, we still have a few issues that we need to iron out. So if you find a bug, please report it right here at the bottom of the page, and feel free to contact us through any of these means.